All right. Hello, everyone. Welcome back to part 2 of our symposium. Our first afternoon speaker is Bilal Sheikh. Mr. Sheikh works as a bioinformatician in Jonathan Carr's lab at the Icahn School of Medicine at Mount Sinai. His interests are in developing tools to enable large-scale simulation and analysis of biology through application of computational technologies, with the ultimate goal of allowing predictive tools that can elucidate molecular pathways and enable personalized medicine. As the lead developer for the Biosimulations project, he's here today to give us an overview. Please give Bilal Sheikh a warm welcome.
Hello, everyone. Thank you very much for the kind introduction. Today I'm going to be talking about Biosimulations, which is a project that we've been working on, which is designed to make running simulations easier. So let me just get started and share my screen here. All right, is that visible?
It's showing presenter view right now.
Oh, so it's the wrong screen. All right. So before I get started, just want to give some acknowledgements to the rest of the team that's been working on this, especially the people in bold who are, the main people on the project, as well as all of our various collaborators. So a quick outline of what I plan on talking about. So first, I'll start off with a quick introduction, talk a little bit about the motivations for the project and the overview of the tools and the platforms that we've developed. I want to spend most of this time doing a live demonstration, showing essentially the platform that we've created to execute biological models and simulations in the cloud so that you don't have to really do much on your own computer. And then if we have time, I'll talk a little bit about the ecosystem of tools and technologies that sort of enable this to happen. That's sort of more geared towards software developers. So I guess we'll kind of poll the audience and see if that's something that is worth getting into. And then finally, I want to give sort of like a roadmap of the next step in this project and some of the upcoming work that we have, as well as where we're planning on going from here. So the rapid growth of technology and the explosion of the biological data that's available has led to really large advances in systems biology and fields like precision medicine and bioengineering. However, further advancements in these fields will require larger and more comprehensive and more predictive models. These sorts of models will need to integrate multiple modeling methodologies and frameworks across scales and domains, and there's going to be a lot of collaboration needed between different teams and different multidisciplinary teams. For example, computational biologists are going to be working alongside experimentalists. People who are familiar with modeling are going to be working alongside, for example, clinicians who are not at all involved in modeling or computation. So in order to really be able to achieve these goals, it really must be easier to share and reuse models and simulation studies across a wide range of audiences. So we do have quite a few existing community standards that are designed to make this sort of interoperability possible. For example, keeping track of things like the different frameworks, different algorithms and parameters. We have some community ontologies. For actually designing and constructing models and simulations, there are languages such as SBML and SBML that we've been discussing, as well as other languages for other domains such as CellML and NeuroML. And like we were going over yesterday, there is a way to sort of containerize all of these into projects through the Combine Archive standard. However, there are still quite a few barriers to reproducibility and reuse. For example, simulation tools support various different algorithms, features, formalisms, and formats, so that can make it difficult to determine which tools to use for a particular model. For example, right now, as we've been working with Tellurium, a question came up earlier about introducing PDEs into the model. And that's something that Tellurium, for example, doesn't support. There are other tools that do support PDEs that then don't support things like spatial models or discrete models, stochastic models. So essentially, there's quite a few different techniques and formats that are available out there, and many different tools that all sort of have different support for those. It can also be quite difficult sometimes to obtain specific versions of tools for older specific models, for older published models. So if you take a look at a paper that's published a particular model and try to simulate it, it might just not work because since then the tool has been updated and things are now slightly different. And finally, it can be difficult to install or use simulation tools. So we've been using Jupyter Notebook so far in the class, which has made it much easier. And Tellurium is actually quite easy to just install because you can do pip install Tellurium. But some of other simulators, for example, you need to have quite a few backend C libraries installed on your computer, different sort of optimization libraries. And these all have to be linked together dynamically. So for someone who is not at all involved in the modeling field because their focus and their expertise is in experimental biology, it can become quite a challenge to be able to download and use these tools. So here's just another sort of look at this problem where if you are a reviewer who is looking at a model that's being published, you're essentially just kind of taking the authors at their word. They say that this is what their model does, and it's quite hard for you to figure out how to run this thing. So you kind of agree. Same thing with reading a paper. You essentially have to treat this model as black box. And for those of us who are just learning how to actually model, this can be quite inaccessible to getting started. Something that's a little bit more difficult, that's also difficult, is viewing actual simulations and simulation results. So we were talking about the Biomodels database where we can go and download models, but we don't really have a central repository similar to that for simulation results. So once you've downloaded a model, if you want to see what the results of that simulation are, you essentially need to run it yourself instead of being able to view the results the same way that you can view a model. Same thing with once this is actually done, you end up downloading a large amount of numerical data rather than anything that is more easily parsable. And it's really up to the user to figure out how to actually look at this data, parse it, visualize it, and analyze it. So the goals of the project then really are to make it easier to share, discover, and understand modeling studies. This includes the models, the simulation experiments and results, data visualization of those results, and to make it easier to reproduce and reuse simulations. And the way we do that is by making it easy to find capable simulation tools for a particular model, making it easy to edit a model and simulation parameters, just tweak a few things, run it again, see what happens, and repaint figures with alternative simulation results. So if there is a paper that has been published a figure, we want to see if we tweak this and rerun this model, how is that figure going to be different? And often these figures are very particularly manually tweaked and curated to make them look nice and publishable. So it can be quite difficult to recreate that figure with different data. And finally, we want to be able to support a broad range of modeling across various methods, biology, and different scales. So we were talking a little bit earlier about community models of various cells interacting down to just small models of individual metabolic reactions, sort of provide a unified interface for these sorts of simulations. So the toolkit that we have been working on, it's essentially here, the goal is to be able to take all these various types of simulations that exist, all the various types of rules that exist, package them into a standardized format, and then put them through our Run Biosimulations platform, where you can then share these results. You can export these results into just raw data, as well as create visualizations to view these results. And so this toolkit is sort of built up on the community resources and quality control that essentially already exists. So like we've been discussing throughout the past week, and like I was mentioning earlier, the community has already created quite a bit of resources to enable this standardization and enable this interoperability. But we are trying to essentially bring all of those together and provide a layer of quality control over that. So just because these standards do exist doesn't unfortunately mean that they are very well adhered to in the real world. We'll probably see later on in the demo that there are quite a few examples where different repositories may have models that do not exactly adhere to these standards. So we're both using those standards and creating this level of quality control on top of those. On top of that, we have worked on a project that we call Biosimulators. It's a bit confusing. It's different from Biosimulations. So Biosimulators is what a registry of standardized simulation tools. So these are tools that have a consistent interface. They are designed as Docker containers, so there's no installation needed on your local device. You can just enter a command into your terminal. They'll be downloaded automatically and executed automatically. And on top of that, then we've created this web application where you can actually execute and simulate various models and then visualize their results. In our backend, what we are doing is using these standardized simulation tools to run whatever simulation is provided by the user. But then on the actual platform itself, that's completely invisible to the user. They don't need to know anything about that. They can just upload a model and visualize it, simulate it, and see what happens. And on top of that, we are now planning on building Biosimulations as a platform. And this is going to be essentially a platform where authors can share their simulations, people can interact with them, create forks of those. And this is still in progress. So forgot to mention earlier, but if anyone has any questions, please feel free to interrupt at any time. Just turn on your mic. I'm trying to monitor the chat, but it's a bit difficult with sharing the screen. So please just go ahead and interrupt. And I guess I'll use this as a chance to ask if there are any questions so far. Cool. Okay. So kind of drilling in a little bit more to that figure that we saw earlier, what we are doing in our platform is that we're using then these standardized simulators to essentially take in our omics archives, create these standardized simulation results that we can then go ahead and visualize. So I'll switch over now to the demo. So doing the things that we're going to really look at are going to be how can we actually execute simulations? How can we then visualize those results? How can we find and execute a particular simulator if we want to look at a model further? As well as a couple of tools that we've created for validating projects and actually putting together these Combine archives. So if you're interested in following along, you could take a look at the website that I'll be using. So we have a stable version and a preview, which is has the latest features. I'll be using the preview. I believe there are a couple of features that haven't been pushed to the stable version yet. So if you want to follow along, you can just head over to run.biosimulations.dev or run.biosimulations.org. Most of it should work. So here you can see the run.biosimulations website. And if we can just to go ahead and get started, we can just go and click on run a simulation. So it'll bring us here to this form where we now have the option of uploading an omics archive or uploading an omics archive that we already have, or providing a URL for an omics archive. So in this case, I'm just going to go ahead and upload one of the examples that we have. Okay, I guess I have not downloaded the examples. And so here we go. So I uploaded an omics archive, and you can see that the platform actually was able to determine a little bit of information about this. So it was able to see that, okay, so this is an SBML file. it is using these particular algorithms here, and we are actually able to support this by running the exact same algorithm that's been provided. We can actually drill down on this a little further if we want. And for example, if there is a model that we are running that is not supported on the platform, we can pick something. Okay, just give me something that will at least approximate it, or give me something that at least has similar techniques and similar variables that it's trying to do. So over here now, we can go ahead and pick a simulation tool. We've been using Tellurium, so I'll go ahead and click on that one. And you can also then pick a version of the tool. So we've only have version 2.2 of Tellurium available, but as Tellurium goes ahead and releases new versions, we will incorporate those here and keep a record of all of the different versions. We can pick some computational resources, doesn't really matter too much for the purposes of this demo, and we can give this a name. So I believe what I uploaded was a cancer model. So I'm going to go ahead and call that humor oscillations. And if you want, you can enter your email address, and that will give you an email notification when the simulation has finished running. In this case, it's pretty quick. It'll be within a couple of minutes, but for longer running simulations, it might be helpful. So I'm going to go ahead and hit run. And you can see it's now redirected us to this page. The simulation has joined the queue, and there we go. It's now started running. So rather than sit here and wait for this, I'll actually go ahead to some of the runs that I've already created. And this is the equivalent run that I ran a couple of seconds ago. So you can see here some information about the project. So if I click on this, it'll download the omics archive that I just uploaded. This was actually run using Pisces, which is a different simulator, not Tellurium. But I can click on the simulator here. It'll provide me some information about what tool exactly was used, what sort of algorithms that tool supports. We'll come back to this in a little bit. Gives me some information about how the run processed, and I can click on this actually and then download the raw results. So this will give me a zip file which contains the raw results of the simulation run, the omics archive, any sort of figures that were generated, any plots that were output. If I click here on simulation project, you can actually see some of the metadata that was already included in the Combine archive. So from that, we're able to get a little image of what this model represents. So this, I suppose, is going to be a figure from the paper it was published in that was included. We have information about the authors of the page, as well as links out to their ORCID profiles, as well as some metadata about what process this is really simulating. And so this isn't from a database necessarily, but this metadata, if it's encoded in the omics archive itself, we're able to provide this sort of information to the users. So from over here, though, I can actually now go ahead and select some of the visualizations that are present. So if you see here, there's this figure right here. So this is actually a figure that was defined in the SBML itself. So if you think about earlier when we were working on these Tellurium models and we were deciding, what the simulation should be, what the plot should be, and then exporting those into an omics archive, that information about how to run the simulation and that information about the outputs has been encoded into that omics archive. So when I select that one and go ahead and visualize the chart, I'm able to get a view of the figure and see what the output was. So if you are, for example, working on creating models like we were earlier, but we want to send them to collaborators who are not at all familiar with Python, for example, or who do not have a computational background, you can export those archives like we did and send them a link to the website where they can then go ahead and execute those. Finally, we do also have a log of the simulation run. So we can see that there was one task here in this model. Here's the algorithm that was used to execute it. Here are some of the parameters that were provided to the algorithm. And then over here, we can look at the outputs that were produced, for example. And this one is actually just a raw output of the entire backend system that we're using. So for transparency, you can see exactly what it is that we're doing here on the backend. We download the archive that's provided, get a Docker image of the container, go ahead and run it. Here's a lot of information, and here we are then updating the results to our platform. So I'm going to go back now to this option here to select a chart. And instead of using the chart that the author of this project provided, I'm actually going to go ahead and create my own. So I can select, for example, some number of curves that I want to look at. And let's just plot these against time. And from here, I can now select some of the variables that are present in the simulation. I guess there's only 3 variables in this case, so it's going to end up looking exactly the same as the chart we just saw. But this time it's custom. We created it ourselves. And we have the option here now of changing some of the scales on the plot. I can look at this as a line plot or a scatter plot, for example. And now as you can take a look at the chart that we've created, it's now sort of tuned to what it is that we're trying to explore. So now I'm going to go back to another simulation, which actually shows this idea of creating custom visualizations a little bit better. So I guess while we're here, we can demonstrate the sort of ability to have various models that you have run, as well as sort of filter them based on various parameters. So what I'm looking for right now is a simulation that I ran through. So I'm going to click on this one. So now you can take this look here from this information. This is a E. coli model. It is a complete flux balance analysis of the entire core metabolism. And it was information here about the framework. Essentially, this is it's a flux balance model. So it'll be different from the ODE models that we've been working on. So if we take a look at the chart here, this includes a custom Vega file. All you need to know about that right now is that it's a custom specification for designing your figure. And you can go ahead and visualize that. You'll see that we have now here a metabolic map of the E. coli model. And all of the data actually on this map, all of these different paths are encoded with information about how the metabolism proceeded in this model that we have. So you can see that higher up on this scale, the more yellow colors here represent higher amounts of flux, so more active metabolic pathways. And down here, the purple are sort of lower activity. And these were calculated from the simulation that we ran. So if I were to, for example, go here to the simulation run and run it, click this rerun button, you can see it's now set up this for me over here. And I can actually rerun this, see if there are, for example, changes that I want to make to the omics file, and simulate it. And then when I come back to that model, I'll be able to just kind of select the chart. Come back to that model, we'll be able to see how this really changed. So we think this is a really good feature because it provides a way for a lot of the publications, a lot of the figures that you see in publications to actually be represented in a way where you can recreate those figures with different data sets. All right. Okay. Just get back here for a second. I can look at my notes. Okay. So let's focus on this simulation here, and we've been able to take a look at this. We've looked at some basic visualization, but we realized that okay, this is something that we really want to explore further. And how can I go ahead and actually download a tool to make it possible for me to run this? So if you go to the list of simulations here, we used to have information about the framework. I don't know if that's. Yeah, so like I said, this is a flux balance model. So sorry, let me just open that back up. So if I go ahead and click on the simulator that this was run with, it'll take me to, like I mentioned earlier, this other system that we're working on called Biosimulators. So here we're really looking at a database, a registry of all the different types of simulation tools that really exist in systems biology. Obviously not all of them, but as many as we have been able to gather. And over here we can see, the sort of metadata about the tool. And what's the important part of this here is this algorithms tab, where we can see the different types of algorithms that are supported by the simulator. So like I said, this was a flux balance model. So if we try to run this with Tellurium, for example, which runs ODE models, that might not work. Unless, correct me if I'm wrong, I don't think Tellurium has support for flux balance models. So we now need to take a look at okay, so how can we actually run this? So if we see here that the tool that was used was CobraPi, and we can take a look at the algorithms that it has available. So it has this algorithm called flux balance analysis, which has a very precise meaning through this ontology. There's flux variability analysis and so on. So now if I go and take a look at all the different simulators we have, I can now go here and say, okay, so I need to run a flux balance analysis model. So let me look for simulators that support flux balance analysis. And here now you can see, these are the various models that are available. And from here, let's say we want to take a look at, I don't want to have to install anything onto my computer. I don't want to have to work out what my environment is. So let me only look at the ones that have Docker images available. So okay, that narrows it down to 2. CobraPi is the one that we were just trying online. So let's try SBML instead. So when I click on this now, I can see that, same sort of information that was provided earlier. I can take a look at the algorithms, see that, they essentially have the same sort of algorithms. Take a look at, what versions there are. So in this case, just 1. And then here is actually just a list of sort of the specifications for the simulator. So everything that we've been looking at, except in a more programmatic form, as well as actually a set of test results. So these are test results that were run with the simulator against a whole suite of examples. So we can see and make sure that, okay, the simulator, for example, is going to produce a report that has, the correct number of dimensions. And it's going to be able to support, simulations that have these various features included. So from here, if we
are the great ones things that you just didn't run at all, or can't run, or don't make sense, or?
Oh yeah, so the ones that are skipped essentially are things that don't really make sense in this case. So most of these are whether or not the simulator can run these particular models from biomodels. And I believe most of these are, well, so here's an SSA model, here is a ODE model, which doesn't make sense to run through a flux balance simulator. So that's why those are skipped.
Got it, thanks.
Yep. So now if I come here to this page, you can see this little section here, which is how to run these simulations locally. So I can actually just go ahead and copy this to my clipboard. And if I open up my terminal now, second. I should have prepped this a little bit earlier. Got to do so. Okay. So here, let me zoom a little here. So here we have a omics file of the archive that we were just looking at online. So I'm going to paste in the command that I copied from the website. And you can see that I currently don't have this installed on my computer. So it should go ahead now and actually download the simulator for me. So just give it a couple of seconds to process. And if anyone is familiar with Docker containers, they are essentially ways of wrapping up all of the dependencies, all of the software that you need for a particular program into one container that can then run on multiple platforms without having to really work on the environment yourself. So we can now see that it's been downloaded. So I can now go ahead and run this. And it'll give me a little bit of a help output that can show me how to use this. There we go. Yeah, so here there is how I'm supposed to use it down here at the bottom. So you can see that I give an archive as input and then provide a directory as output. So I'm going to go ahead and run this again, but this time provide those variables. So this was the E. coli. No, sorry. So one thing that I forgot actually, when you run a Docker container, you need to bind your working directory. So let me just quickly, so typing this out, I'll just try to copy it. Sorry about that. So this is just a little bit of boilerplate that kind of needs to be in here, and this is to allow the container that we are downloading to actually access our local files. If you go on to the Biosimulations website, we have documentation that would cover in more detail how to do that. So now hopefully this works. If I typed everything correctly. This portion needs to be moved further. So now you can see, it's outputting a good amount of information. This is very similar to the logs that we were taking a look at earlier. But what we really want to look at is just right here at this end. So we can see it executed one task similar to what we had done online. And it was able to do it successfully. And now if I take a look at the directory, previously, if you remember, there was just this biomodel archive here. We now have a log that's been output. We now have these reports that have been output. We've got our SBML file from the archive, as well as if there had been some plots generated, we would have seen a folder with plots as well. And now this reports essentially contains all the data that was used to generate the figure online. So now if I want to make some changes to this and say, okay, so I've run this model using CBMPy, what would I need to do in order to run this using CobraPy? And so if I go back now to the Biosimulators database, and let's go back to our simulators, take a look at, remember it was CBMPy and CobraPy that had this correct framework. And if I see, all that we need to do is use this Docker command instead. Use this Docker image instead. CobraPy version 0.22. So I'm going to take the same command that I had earlier and just replace the name and version simulator that I'm using. And you can see that I don't have this image available on my PC, so it'll go ahead and just download everything for me. Since I downloaded the other image earlier, and these sort of have the same root, you can see that a lot of this information was already cached. It can now download and extract that image, and it'll go ahead and hopefully just run it run it the same way. And so we think that this, really provides a set of very powerful tools for, studying and working with these models, because instead of having to download these various programs, figure out how is it that I install them, how is it that I actually use them, we have this now unified interface where all these different simulators can be run the exact same way on various inputs. So essentially all you need to do is put in this command, and this is a little bit that changes from version to version, where I can just put in the name of one of the different simulators that are supported, different version, and it'll run. And this is as opposed to trying to learn a new interface, figure out where's the menu items for this particular simulator, etc. And we can see, if I take a look here again, it has created out the exact same results that we were expecting. So we head back here. So we've been able to essentially execute a simulation online. We have been able to find a particular simulator that we can then use to download and work on things locally. And now we can actually say, okay, so how do we actually create these omics archives? So we had discussed yesterday essentially how you can export them from Tellurium, but there are lots of models that are available online, for example, that don't necessarily have this information. So if we go ahead and get a particular model from Biomodels, let's see if I have this saved. So here is actually the wolf model that we were working with earlier. So I'm just going to go ahead and download the archive that was generated here. And I'm going to go and try to run this. Well, there's no need to run it. I've already run it here. So you can see this is the wolf model that we were working with earlier. I ran it through the system here. And you can see that this one actually didn't give us a output. The run failed. And if I take a look at why, you can see that this Combine archive actually is not valid. So like I was mentioning earlier, we do have a lot of these community standards that make all these tools work together and interoperate, but unfortunately there isn't a lot of validation on those standards. So older models that have been uploaded to biomodels, for example, may actually have incorrect adherence to these standards, and there might be something that causes issues. So when I try to run this, I get an error. And it tells me actually, okay, the Combine archive is invalid, and the reason it's invalid is because this file, doesn't really make sense. This is not a file that actually, exists. So I can now go ahead and try and see, why exactly this didn't work. So I downloaded this file. I can now open that up locally. And I don't know if this was mentioned yesterday, but omics archives are essentially just zip files. So you can, unzip them in the same way that you can just any normal zip file. So now, unfortunately, Windows doesn't recognize that there's a file, so you'll need either a command line or something like 7-zip.
Or you can just rename it to .zip.
That you could also do. That's probably an easier way to do it. So I've unzipped this file here, and you can see that we have our main XML, which is our model. We have, I believe, wolf XML, which is our model, and main XML, which should be the setML. But if I take a look at the manifest, if I take a look here at this manifest, we can see that, okay, so we have a main XML file, a wolf XML file, so the wolf is going to be the model, and then here's our setML. Okay, so let me open up the setML and see. So over here, here's the problem. So here's our list of models. We're saying that we want to run this particular model here, but the source that it's pointing to is wolf_SBML.xml. But if we look at our file, we actually have main.xml. sorry, wolf.xml without the underscore SBML. So this, the system here is essentially able to take a look at these sorts of, errors and actually kind of point you to the right direction and, try and figure out what it is that was going on. So we have actually this.
What's the weird thing with the /temp thing? I assumed that was going to be in the file, but it's not.
Yeah, so this is actually just the directory that it happened to be running in on the back end. So when it tried to run this and actually parse it, it included this.
So it unzipped it to that temp directory,
yeah.
Then looked for the file. Okay.
Yeah. So this is actually a good thing that we can actually fix here, because this error only shows up if it's not a valid file. So we actually never noticed that that subpath there.
Right.
So we actually have a utility function. I don't agree from that. Utilities function here where we can actually, do this without having to run a simulation. So we can either, again, upload a file, point it to a URL, and get this sort of information about a particular omics archive. So in this case, I'm going to upload, we already saw what the errors look like. So let's say I go in and I fix that error, and then I, upload it again and hit validate. I should be able to then get back. Okay, so in this case, we have a couple of warnings actually, but no errors. So, in this case, this might actually be, I've been able to fix this problem and I can now go ahead and try to run it again. So let's say that, there's quite a few errors, not really able to figure out how we can fix this. So I can actually just go ahead and create a simulation project without needing to necessarily parse through everything, and just by using the model file on its own. So over here, we're going to upload a model file. So I'm going to go back to where I unzipped that folder. Download thing. And go ahead and select the model file without the rest of the file there. And I know that this is SBML, so I can click on that. And based on that, it actually should be able to potentially, can it do it? Not necessarily. Sometimes based on the file that you upload, it can actually go ahead and process this for you on its own. But so this is going to be an ODE model like we've been working with. So that's a non-spatial continuous. And I'm going to select the type of simulation I want. I want a time course. Let's go ahead and start at 0. Let's go until let's do 20. This is essentially, so this is the time that you're starting your simulation, but you don't necessarily need to save those results. So this is the time where you actually want to start saving the results. So let's again do that 0, and I want to do 1,000 steps. Over here, we can go ahead and select which algorithm we want to run this simulation with. So this does require a little bit more background knowledge about how simulations work. But for ODE models, generally very, very general, can't count on this. CVOT is a good algorithm that will, work. And here I actually now can see some of the various parameters that exist for this model. For this algorithm. So the CVOT algorithm has, some concepts of tolerance that it uses to calculate things. It has these, leap steps and sizes. And so you can, go ahead and tweak these a little bit, and we'll also tell you which simulators actually support tweaking this. So Tellurium, for example, allows you to change the maximum BDF order. I'm not sure what that is, but if you want to change it, you can. But Pisces, for example, doesn't support this, parameter in particular. And then here, vice versa, for whether or not you should automatically reduce your tolerances over time. Here, we can take a look at the actual various parameters, various species and concentrations that are present here. So we can look at this initial amount of species A3C and see that it's set to 1.5. Okay, well, in this case, let's see what happens if I bump that up a little bit to 1.8. And let's say there's this parameter here that's set to 5. Not exactly sure what it is, but let's just tweak that to 4.5 instead.
Hi, Bilal.
Yeah.
If you could wrap up your talk within the next
yes,
definitely. So we can have questions.
Thanks. And then from here, this is essentially just what we want to output. So if you remember from plotting our Tellurium simulations, from plotting the simulations on Tellurium yesterday, we can kind of pick the sort of species that we want to display. And then this will give you a little bit of information about, okay, these are the tools that you can run this on. And if I just, hit download here, you'll see that it's now giving me a zip file, which essentially an omics archive that I can then now, use to upload and run simulations here, or I can use this to load them into Tellurium the same way that was demonstrated a couple of days ago and use it there. Or send this to a colleague who wants to see how my model works under some particular conditions. If you wanted to, you can also just hit simulate here, and instead of downloading it and uploading it back up again, it'll directly take you to the page where you can run this directly. Yeah, so with that, we are essentially done with what I had wanted to demonstrate. I guess we're short on time, so we won't go through the way that this is sort of all built in that much detail. But essentially, what we have done, like we were talking about, these sort of, in purple here, these standards kind of already exist. So on top of these, we've built these validation tools that allow us to make sure that the standards are adhered to, that they're all going to be working with each other. Then we have used Docker to create these simulation tools, which are all following a standardized format and input, which allows them to be used in a similar way and a consistent way to all the others. And this then, when you take these simulation tools that we've containerized, give them a simulation project through a Combine archive, are then producing results in standardized formats that we can then build on top of to visualize, explore data further, parse those logs and understand what went wrong, what worked, what didn't work. And this is sort of the pipeline that enables all of this to go ahead and process. So very quickly, the results are produced as HDF5 files, which are essentially hierarchical files. Think of pandas data frames, but on much, much larger scales. You can have multiple terabytes, and you can label each of those data sets with attributes. So that's how we're able to pick the particular variables that we want to be plotted, for example. The logs themselves are in have a JSON schema. So if the simulation tool outputs this JSON schema, we are able to parse them and understand the various steps. Each of the tools has been, the capabilities have been recorded through a lot of standardized ontologies. We've gotten some metadata about them, and we've verified them by running test results that enables us to search through and say, okay, which simulation tools support which sort of algorithms. And finally, for the visualizations, we're using Vega to provide these sorts of figures that can be replicated easily and painted with new data, as well as just some basic interactive plots, just through standard plotting libraries on the web. And let's see, yep, it's just some more information about the Docker tools that we're discussing. Yeah, so most of this, like I mentioned, was from a much longer presentation targeted at developers. So let's skip through most of that. Yeah, so in terms of the roadmap for our project, so like I mentioned, we've really this foundational resources were all built before we got started on this, through the community over the years. We have created biosimulators, which is the registry of all these tools. These numbers have actually been updated since then, but they contain a lot of standardized tools that you can go ahead and download and run, sort through. Those are all working. We have run biosimulations, which is what we had just demonstrated. Again, this is already out of date, so we now can support models and results of essentially unlimited size within reason, and can provide various different visualization modes. And the next step that we're really hoping to work on is the Biosimulations Platform, which will be a way where you can actually go and browse all of the different simulations that people have run, and take a look at those. So right now we saw that list of simulations. Those were all simulations that I had uploaded or loaded from the test options that are the examples that are provided. But in the future, we really hope to make a platform where an author can go ahead and publish the simulations that they were working on, send a result to them, their colleagues via URL, explore the simulation, and really create a platform for collaborative simulation and modeling building. So finally, if you want to connect with us. All of our work happens very much in the open, transparently on GitHub. We submit any sort of issues that we have there. Our features and ideas, our discussions are all publicly available. So if you want to get involved, all you have to do is head over to our GitHub, take a look at the discussions, and you can go ahead and comment or follow along, post any issues, or if you have a particular simulator, for example, that you want to use that's not available, you can submit that on GitHub as well, and we'll take a look and see if we can include it. Yep, thank you very much. And stop sharing my screen here, and then we can see if there are any questions.
So I just want to add that this is the flagship project for the Center of Reproducibility. So we're very excited about this project. And at least the immediate benefit will be to the community is that you'll be able to take almost any model from biomodels and it'll be able to reproduce pretty much exactly what the paper reported. So hopefully that'll be in place in a few months. So Lucian, Jonathan, and so on are busy curating all those models and biomodels to make sure that they reproduce what was claimed in the paper. So that will be a very exciting development where it'll be a place you can just go. You find a paper you're interested in, you just go there. If it's available on biomodels, you'll see it there, and then you'll be able to run it and do all the things that were shown, and you'll be able to hopefully get the same curves that were reported in the paper.
And it should be integrated into the Biomodels website as well. So you should be able to just go straight from Biomodels to the biosimulators.
Okay, yeah.
Yeah, hopefully there's going to be a little button when you look up a model to just click it and simulate, so you don't even have to download it and upload it again.
So this is the first resource of its kind like this. So we're hoping that other domains other than systems biology may follow suit to make all computational models available in a reproducible manner.
Any questions? I guess I'll turn it back over to the hosts.
Actually, are there any publications you can send people to? I don't know if you mentioned any publications. I think you have one, don't you?
Oh, yeah. So we do have the paper on sort of the platform.
Yeah.
If you want to look at for more information.
It's in nucleic acid research, I think. Is that right?
Yep. So I just posted the link in the
there you go.
Chat.
Good. Oh, yeah,
so if you want some more information about the way it's structured, you can take a look at that. Or the source code for this is available on GitHub, so you can see exactly how it's built. It's all open source.
Which is easier, reading the paper or reading the source code?
Almost certainly reading the paper. And the slides, if you want to take a look at the rest of those, the ones that were skipped over, I'll post those to the same shared folder for all the other resources.
Great, excellent, thank you.
Okay, thank you, Mr. Sheikh, for your talk. We will now be moving on to our next speaker, who is Dr. TJ Sago. Oh, let me. Okay. Dr. Sago is currently a postdoctoral fellow in Professor James Glazier's lab at Indiana University, Bloomington. His background is in mechanical engineering, and his current research involves developing computational models and simulation tools for biological and biomedical applications. Today, Dr. Sago is here to talk about software packages, Mechanica, and CompuCell 3D. So please give Dr. Sago a warm welcome.
Well, thank you very much. Thank you, Herbert, for inviting me to speak today. I'm very excited to be here. Yeah, I am here to talk a lot about space for the next hour. So I imagine that a lot of the modeling that's been done this week has not been particularly keen on a lot of spatial detail and a lot of very fine resolution concerning individual cells. And so I'm going to share my screen. Can you see that okay?
Yep.
Great.
You can see
Great.
it.
Okay. And yeah, so I am a postdoc at the Biocomplexity Institute in James Glazier's lab. I've been working on CompuCell and CompuCell-based modeling for just under 2 years now. And then I've also recently taken over the Mechanica project as well. I'm going to sort of do a survey of both of those for the next 45 to 50 minutes or so, and try to leave a little bit of time for discussion. CompuCell is a cell-based, spatiotemporal, agent-based modeling and simulation environment. CompuCell provides the ability to implement cell-based models that you may develop and would like to share with people and test. And you can simulate these with lots of supporting features, lots of built-in computing capabilities and modeling capabilities. And yeah, you can get a sense of what these sorts of simulations look like here on the right. So we distribute CompuCell with 2 GUIs that support model development, implementation, and then execution. This one here on the right, this is what we just call player. Player is an interactive simulation execution GUI. So you can take a model that you've developed or someone else has developed, you can load the simulation scripts and play them interactively in real time, generate plots, pause, rewind, so on and so forth. On the other hand, the other GUI that we distribute is what we call TWEDIT, which is a supporting GUI for actually developing models. So TWEDIT comes with a lot of built-in model development support features like code snippets, simulation script generation, and so on and so forth. CompuCell, the backend is written in C++, whereas more of the user-facing side presents a Python interface so that users can write their own step-by-step model implementations on the basis of individual cells or groups of cells by type, by location, and at each simulation step. And so TWEDIT supports a lot of that model development because there's a lot of functionality. This is just a brief list of some of my personal favorites here on the left. And TWEDIT sort of boils that down to pointing and clicking on buttons and links and so on and so forth to help you generate those models very quickly and more focus on the biology as opposed to the actual coding. So we distribute CompuCell with a whole host of features, including a PDE solver suite. So there's built-in reaction diffusion solvers that are written into the C++ backend. You can see some of those here. So these are 3 different fields in this particular simulation. We've done a lot of work with CompuCell on integrating additional simulators, including libRoadrunner, and we support model specification in Antimony, CellML, and SBML, where it's possible to take, say, an ODE model that's been specified in Antimony, as if it were written in Tellurium, and then create instances of those model simulations and attach them to cells or attach them to a spatial domain to be able to do things like multi-scale modeling, say with an ODE model that describes intracellular processes that can then be attached to individual cells and coupled with its conditions, as well as, say, taking an ODE model of more of a systemic response something at a much higher level than something like a cellular domain to model something like, say, whole body response, or some response that's going on somewhere outside of a simulation domain that you may be modeling with explicit spatial resolution. So CompuCell also includes a whole battery of modeling components for modeling individual, like, separate processes and properties associated with cells and things that cells do. I've just included a few here to give you a sense, doing very basic things like imposing volume constraints on individual cells, or surface and shape constraints. There's also things like modeling adhesion on the order of pairs of cell types, or individual cell conditions, like cadherin expression. There's also model features to impose external forces, to break up cells into individual compartments and simulate them as a cluster, to get a sense of heterogeneity within cells. And that's just to name a few. Now I wanted to kind of give you a sense of some of the recent work that we've been doing to kind of convey the way that the CompuCell based modeling tends to be used in actual applications. Last year, when the pandemic really hit the States, some of us in our lab wanted to make some sort of contribution to applying our skill set in computational modeling and cell-based modeling, multi-scale modeling in biology, sort of add that into the mix of research that was going on and continues to go on concerning viral infection and immune response. And so that resulted in this paper that we're sort of glancing at here. In particular, this is a modular computational framework that we developed in CompuCell to be able to support large research groups, interdisciplinary research groups who may be working on different aspects of all of the complexity to do with viral infection and immune response, and to be able to work on those sort of sub-projects concurrently, all while contributing to the same sort of framework. And so maybe you can get a sense of that by looking at this schematic of models here. So even with the development of the framework and the preliminary models, there's so much complexity to do with viral infection and immune response that even a sort of early stab at a working framework generated a lot of different models. Things to do with internalization of virus, signaling by cells to invoke an immune response, the actual activities of immune cells whenever they're at a site of infection, so on and so forth. But what was possible is to take those individual models and break them up into modules. And so each of these different aspects of viral infection that we were interested in representing for this framework development, each of those are represented here, and they're all broken up into individual model modules, each of which are executable within the broader framework, as well as interconnectable according to an API. And we do that by leveraging the Python interface for CompuCell model development. In particular, the model specification using Python in CompuCell is done using a particular class, a Python class that's in core CompuCell. And users just will derive a new class based on that one that implements their model. In this case, for a modular sort of modeling framework like this, we have lots of different implementations of that base class, and each of those implements one of these different models. Now, a lot of what's cool about doing this sort of modeling, not just for this application in viral infection, but I think in cell-based modeling in general, is to be able to cast what's typically considered in more of a homogeneous scenario into a cell-based context. And what I mean by that is that there's lots of work that's done to do, say, ODE-based modeling of intracellular processes, gene regulatory networks, and so on and so forth. What's fun about this is to take that sort of model development work and then place those models in the context of a multicellular dynamic system. So it's, one thing to work on what happens inside of an individual cell when it's exposed to a particular set of conditions. It's another thing to then take that model development and put it in a context where the environmental conditions of a particular cell can change. And that cell can also contribute to changes in their environmental conditions. What this looks like in this application was we developed an ODE-based model in Antimony of viral replication. And that looks like here on the left. So this is the whole viral life cycle, starting with extracellular virus that's diffusive around the cells. It gets into a cell through internalization, and that kicks off this ODE model with these 4 equations shown here, in particular with exponential amplification of virus inside of individual cells during replication. That goes through a few more steps and eventually is released back into the environment to do more infection. Now, we can put this model, and did put this model, into every epithelial cell in the simulations of viral infection that we did in this framework. And then started asking questions like, what does a therapy, something like a viral inhibitor, do for the progression of infection during our simulations? And in this case, we were interested in the trade-offs of having later treatments and stronger versus weaker treatments. In our simulations, we started by just taking one individual epithelial cell in an epithelial sheet and putting some virus in it, starting it as infected, and then giving an immune response the ability to try to fight that infection through various mechanisms as viral replication occurs, viral release occurs, and then subsequent infection of neighboring cells. What you see here on the right is a parameter scan introducing antiviral treatment at varying times after initial infection, and also with varying potency. What this looks like in the actual implementation is to take the ODE model and then say at some point in simulation, the therapy occurs, and the therapy's effect is to reduce the exponential amplification during the viral life cycle. In the simplest case, we simply said that at some point during simulation, this coefficient R sub max changes due to the action of the therapy. And this looks like this plot here, in that at our therapy time, this Rmax coefficient is uniformly reduced by some certain factor in every epithelial cell that's alive, whether or not it's infected. And yeah, you can see the effects in this plot here, this grid. So moving from left to right, these dotted lines show the moment in time for each of the sets of experiments when the therapy was applied, where in the leftmost column, the simulation is initialized with the therapy applied, and then in the far right column, much, much later. And then on the top row, we have something like an absolute treatment, in that when the therapy is applied, Rmax is set to 0 and replication stops. Whereas down here in the bottom row, you can see by these coefficients, this is the reduction by which the Rmax coefficient is reduced to. In that here for 0, what this means is that when the therapy is applied, Rmax is multiplied by 0 and shut off. Whereas in the bottom row at 0.875, you see there's a 12.5% reduction in this column. And as you would expect, early treatments here in green, very effective. So we're looking at number of uninfected cells for 10 simulation replicas of each of these scenarios. And yeah, in the green plots for early and strong treatments, not much change in the uninfected cells, not much infection. Whereas when we go very late and very weak with our treatment, as shown here in the bottom right, and then all of these reddish shaded plots, very ineffective. I wanted to just sort of whiz this one by you, you know, given that there's been a lot of ODE modeling in this workshop. This is another sort of mini project in this paper that I thought was really interesting and very fun to do. So we wanted to sort of demonstrate what it looks like to actually pull a module out of the framework and replace it with something else. One sort of impression that we didn't want to leave was that our framework was only relevant for modeling COVID, when in fact, all of the models associated or parameterized to COVID data are totally replaceable. And so we wanted to show this by substituting out the viral replication model that we had calibrated to COVID data and instead replace it with a popular model of a totally different virus. And that looks like this here on the left. So this Hcv model developed by Alan Perelson and some of his colleagues, you know, this is a fairly well-renowned model of Hcv, or at least so I'm told. And this model, to my knowledge, has only always been used and studied or used to study Hcv in the context of an individual cell exposed to some particular environmental conditions. And so this activity here is to take that Hcv model that has been used in those sorts of like static conditions and instead replace the COVID viral replication model with it and do the same simulations. So this is the Hcv multi-compartment viral replication model, but being simulated in the multicellular context of starting with one initial infected cell, having the spread of virus due to viral replication and release according to the rest of the framework modules, and then the corresponding immune response as it was in the original simulations. Interestingly, there's a lot of variability in these simulations. So we take the exact same environmental conditions, exact same initial conditions, but because of the stochasticity that's involved in these sorts of cell-based models, we simulate lots of replicas to see if our system is mostly deterministic or if it presents a lot of potential for variability in outcomes. And you can see in this case for this Hcv ODE model, we simulate 10 simulation replicas, and we get everything from complete infection of our tissue patch in 12 days, all the way to almost no infection at all, which is really interesting and kind of, in my opinion, a very interesting way to think about these ODE models. You know, in this case, for the intracellular modeling, there is a lot to study about an individual cell and a lot that we don't understand by themselves. And putting them in a multicellular context, I think provides, you know, a new way to study and develop these sorts of models by also considering what sorts of contributions they have to the overall emergent complexity that comes from a collection of those cells. Now, again, CompuCell, because the user interface is in Python, presents a lot of capability that's just by virtue of it being in Python. And so, as I said a couple of slides ago, you know, it's not only feasible, but shown here that it's completely possible to develop these sorts of frameworks. Again, not just for the application in viral infection modeling, but rather for applications that require interdisciplinary expertise in model development. It's possible to create these frameworks that can support lots of input from lots of different fields, all working on sort of sub-projects of much larger, complicated problems like the immune response or like viral infection. And so I wanted to show you sort of what this looks like just in a very simple application that we developed, or sort of example that we developed for this paper. So we imagined that there's one group who had been using our framework and they wanted to implement a model that we didn't, which was recovery. So it's shown here in this top row, which is labeled simple. This first group wanted to introduce the ability for the tissue to recover and go back to some sort of totally healthy state, assuming that viral infection was eradicated. And then after that group did their work, then another group came along and thought that they were interested in doing recovery as well, and they didn't like the modeling work by the first one. The first group, had the inclination to implement recovery, but the second group think that they can do it better. Or they just have a disagreement how it's done. And so what this looks like in terms of this framework and really just in CompuCell in general, is something like this. So the Python class that users implement models in CompuCell, they're called steppables. We call them steppables because CompuCell will take the steppable that's been implemented by a user and will inject it into the CompuCell core along with everything else and step it. Every simulation step, there is a function on this steppable class called step that is called at every simulation step, hence steppable. And so in terms of a framework like this, then what's possible to do is to break up every model module into a steppable implementation and define ways that those steppables can interact and share data and so on and so forth. Or in this case, it's possible for one group to come along and inherit, to incorporate the work that's been done by someone else in their implementation and replace the things that they don't like or would like to improve. In this case, the first group defines a few different functions here, one being a criterion for when a dead cell is replaced by an uninfected cell, and then an actual implementation of what that looks like. So what does it actually mean to put a new uninfected cell into the simulation? And the second group, they just decided that they would like to improve the details about the actual recovery criterion defined by the first group. So here what the second group is doing is that they're actually taking the steppable defined by the first group and using everything according to the first group's work except the recovery criterion. So this is all of 20 lines and then just loaded into CompuCell. No problem. Now, on the other side of the scale, a lot of work that we've been interested in has been to develop cell-based models, like here shown for influenza, develop those based on previous work that's been done using ODE models of population dynamics. In this case, this is a project that we worked on with Erica Mochin and Bart Ehrmantraut at Pittsburgh. And back in 2015, they developed this very, very complicated model of influenza infection and immune response. And this immune response spans everything from very early innate response with release of like tumor necrosis factor, all the way to production of specialized antibodies, specialization of B cells, and so on and so forth. And then managed to calibrate it quite well to mouse model data. These simulations, this is what sort of a demonstrative example here. This is a 1 by 1 millimeter patch over the course of 2 weeks. And this model consists of 20 ODEs with 90 model parameters. And the activity in part for this project was to actually generate this spatial model and cell-based rules or interactions about diffusion equations and how they interact with each other, derive all of those based on the original ODE models in a consistent way mathematically, and then implement them in a computationally accurate and efficient way. One of the reasons to do this that I think is very interesting and also promising for a lot of future work is that when you start to add details about space to models of population dynamics that treat a system as being some notion of being homogeneous, often you necessarily have to introduce new models that are completely neglected by the original ODE model. In the case of something like viral infection and immune response, certain immune cells will induce apoptosis in infected cells through contact-mediated interactions. There are ways to write those interactions in a homogenized sense for a set of ODEs. However, when you actually go to consider contact-mediated interactions in a spatially explicit model, it then becomes necessary to start asking questions like, well, how do those contact-mediated interactions actually occur? And not in the sense of like a biomolecular context, but more in the kind of obvious sense of if two cells have to be in contact for an interaction to occur, then what all happens to bring them in contact? You have CD8 plus T cells that migrate all over the body and arrive somehow at, say, an infected cell, and then through contact-mediated interactions induces apoptosis. And so even in the case of a model like this that only considers a tissue patch with lots of systemic responses that represent whole body immune response, there's still model development to be done just to describe the translocation of those immune cells to actually find infected cells, and do that in a way that the spatial model, in certain conditions at least, can actually reproduce representative results consistently with the original ODE model. And so for this work, we're interested in what sorts of spatial aspects about infection that are neglected in the influenza ODE model, what aspects would actually cause the spatial model and the ODE model to strongly disagree. So in our preliminary work, we found that yes, we could generate this spatial model and we could simulate it in a way to where the spatial model results when plotted, with scalar metrics like population numbers and total, like a volume integral of a diffusive field over the entire domain. Yeah, we could generate spatial model results that are indistinguishable from the original ODE model. And then the question that I think was more interesting is what would make them different? And we found a lot of ways. In this very interesting case, in my opinion, we found that initial conditions of infection would cause potentially huge differences between what the spatial model and the ODE model would predict. In this case, we found that when we start with initial conditions where cells are initially infected, in that they're designated in the infected state where they're releasing virus, yeah, the spatial model and the ODE model agree quite well. However, in the original development of the ODE model that they were actually calibrating to, Mechanica, they worked with the scenario of exposure to initial virus, not some predetermined amount of cells that were actually infected, but rather something like taking virus and dumping it into the mouse. And we found that when we simulated those conditions, as you see here in this figure on the right, hugely different. These first two columns reproduce the initial conditions for the two scenarios that Bard and Erica calibrated to. One scenario is shown here with these black lines. The black lines in these plots are the original ODE model. The colorful plots are simulation replicas. In these first two columns, the first was a non-lethal scenario, a lower exposure to virus, and the mice survive. And then you increase the viral exposure by a factor of 10, and in the mice, you find that they die. We find that in the spatial model, when we use those same initial conditions, we have to increase the amount of virus by a factor of 10,000 before we can get any simulation replicas to produce any sort of lethal outcome. Whereas, a factor of even 1,000 still get significant infection, lots of death, lots of immune response, however, recovery in every single simulation replica out of 50 for each of these sets of initial conditions. And it's an interesting question to do this because you think of all of the assumptions that one has to make when developing any of these models, whether it's a spatial cell-based model or it's an ODE model of population dynamics. When introducing these spatial aspects, can then start to ask questions about the original assumptions that were made for the ODE model and which of those assumptions are actually relevant. And in this case, what we hypothesize is that the localization of virus that's neglected in the ODE model is actually quite significant, in that it takes much more virus in a local patch of tissue to actually generate something like widespread infection and a potentially lethal outcome. That's not something you can ask with a homogenized model. So I told myself I was going to keep this as high level as possible, but I also wanted to at least make this available and show you a little bit of how this actually looks. So one thing that we do is CompuCell is available remotely via NanoHub. We have the entire CompuCell framework available just as a general simulation service. We also, though, pull up the chat, try and click and talk at the same time. We also develop more educational tools. And I put a link in the chat, and that is to a tool that corresponds to this particular simulation. It's one of my absolute favorites that got me intrigued about cell-based modeling in general. And that is CompuCell-based modeling work on vascular network formation. So one of the very cool things about doing cell-based modeling in the way that's done in CompuCell using the cellular POTS model is you can take individual model mechanisms, say like factors affecting the shape of a cell, factors say affecting cell chemotaxis, and then you can combine them quite easily and begin to do things like interrogate hypotheses about what are the responsible mechanisms for emergent phenomena that we see in tissue during development, during disease, so on and so forth. In this case, Roland Merks and James Glazier and a few other members of the Biocomplexity Institute at the time, they were interested in the mechanisms of vascular network formation. And what they found was that they could, aside from some standard models like volume, they found that they could generate vascular networks with the combined mechanisms of elongation of cells and contact inhibited chemotaxis. And by that, for those who may not be familiar, contact inhibited chemotaxis would be chemotaxis of cells along a chemical gradient, but only into available space. So whereas ordinary chemotaxis, one cell may push up against another if it's sensitive to some present chemical gradient. In this case, chemotaxis along a gradient only occurs into the extracellular space. And so this was published back in 2008 in PLOS Comp Bio. I reproduced this and then published it as a tool. Again, the link is in the chat. If you don't have a NanoHub account and have to do the registration process and don't want to worry about it, no problem. If you do, it takes about 5 seconds to actually click through it. So I'm gonna, sorry, 1 second. There we go. All right. So this is CompuCell on NanoHub. And this is Roland and James' model for a smaller population than what they simulated. And in particular, so this is Player, as described earlier. You can generate lots of additional graphics rendering and things like that. In this case, we have one chemical field that's being released by these endothelial cells. And you'll notice here, there's this window called the steering panel. This is a way to interactively change configuration details about the models that are implemented. So these just get written in Python as well if you'd like to make these things available, or say do parameter sort of variations on the fly. In this case, these affect the model mechanisms of cell elongation and chemotaxis. So in the first model for elongation, there are 2 parameters, one being some length constraint along the primary axis of each individual cell. I'm going to turn that all the way up. You see there's no effect, and that's because there's a second parameter, which is the magnitude of this constraint, and that is shown here as lambda length. And that's currently set to 0, but you'll notice when I turn this up, now you see the cells start to elongate. You can make this pretty severe. And then secondly, the strength of chemotaxis. So this lambda chemo is a model parameter that affects how sensitive these cells are to the gradients that they are themselves producing. Currently, they are insensitive, but we find that when we turn that up, start to see networks form. You can see those starting up right here. Eventually, this will produce one network that's very dynamic due to the lack of an extracellular matrix, any sort of basement membrane. Those model mechanisms are not included here. And so this network will continue evolving, never really reach a final state. However, as you see this starting to form, this will certainly produce those sorts of networks as published here by Roland Merks and James Glazier. So if you want to try that out, maybe later, you can grab that link and play with that. We have a number of other tools and things like that. So if you're at all interested, I'll have my email at the end. I'll just put it in the chat right now as well. I'd be happy to provide some links to some of those other tools. Okay. Now, one of the other things that we've been doing has been to sort of keep going with this capability of doing multi-method simulation in a multicellular context, and to take that into a different realm of the types of models that CompuCell supports. So CompuCell has supported ODE-based modeling in some form for, well, Herbert would know, this is before I started working on CompuCell. And then we introduced Antimony shortly, I think maybe 2 Septembers ago. So lots of ODE-based modeling in a multicellular context. What we've done very recently is extend that to also support simulation of Boolean networks. And I don't want to get into the details for anyone who isn't familiar right now, though I'd be happy to share resources later. But in general, Boolean networks describe the sorts of networks that ODE models can be developed to describe, but rather than using continuous model variables, instead, each node, something like a variable, each node in a network can either be high or low. And this formalism has been used to describe quite a few, interesting cell behaviors, and irrespective of one's opinion on, whether or not to use them, they have done some interesting things, and lots of people use them. And so what we have done in this project is to take a well-developed simulator of Boolean network models called MABOS and do the same sort of support for the ODE models, but with Boolean network models. So MABOS, in our most recent release of CompuCell, MABOS is now supported with the same functionality. So users have the ability to take their Boolean network models of intracellular processes and then attach them to individual cells, connect them together, and have them interact through contact-mediated interactions and so on and so forth. And that looks something like this. So took a well-known model of the cell cycle according to a Boolean network model, as shown here, and did a little bit of model development to describe how cells affect each other's cell cycle through contact-mediated interactions with delta notch signaling. And we get something like this. So this is something like wound healing with recovery due to expansion via proliferation in the cell cycle. So in these simulations, these 5 plots here are showing the high and low states of some of the nodes of interest in these networks, red being true or high, blue being false or low. And then here in the top left, this is showing the more of the emergent phenotype of these cells, where the red cells in the top left are notch expressing signals, green cells are delta expressing signals. And that is known to be coupled with the cell cycle. And so you can see different spatially resolved aspects having to do with that, like expansion during mitosis. You can see the sort of characteristic feature of delta notch signaling with patterning, where delta and notch signaling will have to resolve to where you don't have 2 delta expressing signals or delta expressing cells in contact. And yeah, doing this is pretty straightforward, again, by virtue of Python and some of the user support features that we developed to try to make this as biologically focused as possible. I'm just going to skip this because I could talk for days about how cool I think this is to take both the ODE and Boolean models and put them in a multicellular context. Probably the point has landed, so I'm just going to keep going and reserve a few minutes for Mechanica. So Mechanica is a much younger project. CompuCell has been developed since I was a teenager, whereas Mechanica is much younger. CompuCell is a lattice-based methodology, so we start with a predetermined set of coordinates and say that cells can occupy certain sites. Mechanica is an off-lattice particle-based simulator. Mechanica, here's an example of what this looks like here on the right. So Mechanica is a particle-based off-lattice method that's like CompuCell written in C++ and Python. Mechanica is very fast, in part by virtue of that it is a particle-based and off-lattice method, and then also by virtue of that it is GPU accelerated. Lots of performance improvements that were developed by my predecessor, Andy Samoji, when building the early work of this project. This is available on GitHub if you're interested, as is CompuCell. I don't think I included the link, so if you're interested, please just email me or I can put it in the chat in our last few minutes. So both projects available on GitHub. CompuCell is available at compucell.org, whereas Mechanica, there's an older version that's available via pip. One of my current projects is making a conda distribution available. So for those who may be interested, here in the very near future, command away from your terminal to be able to actually run this and play with it yourself. Now, Mechanica can already do quite a lot. So Mechanica is quite unique in a lot of ways in that it can do a lot of things for a particle-based simulator. Again, Mechanica is written in both C++ and Python. The more common model specification would be in Python. And in Python, it's possible to do creation and destruction of particles on the fly for things like cell death or simulating mitosis. There are already a lot of ways to describe how particles in a simulation, whatever they may be representing, whether it be an individual cell or a component of a cell, there are a lot of ways to describe the way that particles interact in Mechanica, whether they be through explicitly described forces or using potential functions that describe the interactions among cells by pairs of types of particles. It's also possible to assemble particles into larger structures using what are called bonds. Something like a deformable rod with mechanical properties that join two particles and describe the energy required to displace those particles with respect to each other. Mechanica already has a primitive event system for event-based modeling. So, for example, describing events that occur at a particular time. One of my current projects is being able to describe things that occur according to other types of events, like say when cells come in contact, when cells are exposed to, say, particular concentrations of a diffusive field, so on and so forth. There are methods that are implemented to do things like diffusion, interparticle diffusion. So actually simulating something like a continuous field, but being discretized according to the particles available in a simulation, using flux descriptions of quantities of species that migrate to and from individual particles in a simulation. In the very simplest case for a particle-based formalism, you can describe a cell as an individual point in space with biophysical and biochemical properties. This is often referred to as a center model. So rather than doing something like in CompuCell, where we actually simulate model or volume exclusion, instead we say that a cell approximately occupies a point in space. And if two cells come close to each other, they may do something like violate each other's implicit space. And we handle that by having things like repulsive forces that bring about the idea of volume exclusion. So two cells can't sit on top of each other, neither should two particles be able to for a center-based model. And so we use potential functions and forces to be able to implement those sorts of ideas for some biological realism using a computationally efficient particle-based approach. Now, what I think is really promising about this approach and the way that Mechanica has been built so far and the direction that it's going now is the ability to construct more complex objects from much simpler objects like particles, like something very elementary like a particle and a set of well-defined and simple interactions between them that then constitute some more complicated structure. You can think of an abstraction of like a membrane as consisting of sets of particles that define surface segments. And when gluing those surface segments together, you have something like a sheet. And you connect that sheet continuously and you get a membrane, and you can define subdomains within a space. You can do the same sorts of abstractions for lots of biological components that make up a cell. And I think this is probably what I'm most excited about with this project, is to be able to take individual subcellular components that constitute a cell and cast the properties and the behaviors of those individual components in terms of these elementary interactions and construct something like a particle-based cell. So a deformable membrane, actual organelles. You can imagine creating something like this helical structure here by just assembling particles with bonded interactions, and use those to describe the cytoskeleton, describe the way that those individual structural components form, the way that they're broken, and how they contribute to the biomechanical stability and dynamics of the cell shape.
Hi, so if you could wrap up your talk in the next few minutes, just a reminder.
Yeah, great.
Thank you.
Yeah, so as it turns out, this is the last that I have for us today. I just wanted to show you this very quickly, a very simple demo of cell sorting with Mechanica. So this is what this looks like. Again, this is written in C++ and Python. This is writing a simulation for cell sorting. In Python, in particular, you'll notice that there are two types of cells. So in very classic cell sorting, you have two cell types in an aggregate, and through differential adhesion interactions and motility, you'll find that they will group together by type. So they'll sort out, and in a lot of scenarios, you'll find the more adhesive species at the core of an aggregate and the looser adhesive along the perimeter. And so Mechanica describes particle types using a class definition. This is called the particle type. And then it's quite easy to do things like create instances of potential functions and describe them in terms of model parameters that define the potential function, and then what's called binding them according to the biological or the model objects in a simulation. In this case, taking a Moore's potential with different model parameters and then associating them as interactions between pairs of particle types, A to A, B to B, A to B. And it looks like this. And I'll conclude with this. We have 6 minutes left for questions and discussion. So I'll leave it at that. Thank you again. I'd be happy to answer any questions with what little time we have left.
Thank you for your talk. Does anyone have any questions?
There's one question from the chat that was, are there different cell types with different models inside them in CompuCell?
So the way cell types are cast is just in terms of categorizing populations within cells, and then providing the availability of taking those categories and doing modeling according to it. So there is, I would say there's not something like a standard built-in definition of like an epithelial cell. However, CompuCell supports being able to specify an arbitrary number of named cell types and then assign those types to cells in simulation and do modeling, model parameters, interactions according to those categorizations.
And different internal models then also, like you could have one cell ML model, one SBML model for one cell type and a different SBML model for a different cell type. Yeah, okay.
That's right. Yeah, although to be clear, for those models, we take individual instances of libRoadrunner and attach them to every cell that we're interested in doing that modeling. Yeah. So in a lot of cases, we end up having, you know, a few to tens of thousands of instances of libRoadrunner running in the background.
So in some sense, technically there's tens of, like every cell is its own SBML model, but
That's
they're
right.
all initialized, like every cell of a given cell type is initialized from the same initial state of some SBML model, and then it just goes from there on its own. Yeah.
That's right.
I'm just curious about the sort of challenges you had with coordinating and like, I guess, the message passing that you would need to do between these cell types, if that.
For CompuCell or for Mechanica?
For CompuCell.
Yeah, I mean, it's pretty involved. Although, you know, and if I understand correctly the history of CompuCell, it has tended more towards simplicity of software for development's sake, as opposed to complexity for performance sake. I mean, you know, we use OpenMP, and it's multi-threaded. GPU acceleration tends to only be relevant for the diffusion solvers because of the formalism that's used for the cellular dynamics. But we also find that in the overwhelming majority of cases, the diffusion solvers are the overwhelming majority of the computational cost. So the diffusion solvers are typically GPU accelerated, although that functionality is down right now while we upgrade. And in that case, it's actually fairly straightforward. Yeah, doing diffusion on GPUs isn't too bad.
You have to sort of do like figure out what your time step is when your differences to coordinate between the diffusion and the ODEs in the cells.
Yeah, well, so the diffusion solver suite does automatic time stepping. So part of the simulation specification does not include any sort of explicit units about time. Instead, the model specification is cast in unitless terms, and then the diffusion solvers will do sub-stepping according to unitless stability. So for every type of lattice we support, you know, there's a corresponding maximum diffusion coefficient that's stable, numerically stable on that lattice, and then the diffusion solvers will do sub-stepping to maintain that stability. In which case, you know, coupling with the SBML models isn't too bad.
So it's all, so the modeler doesn't have to worry about that part, basically. It's all sort of handled under the hood by, it sort of automatically checks things.
Yeah,
Nice.
the legwork on the user's end is taking, you know, whatever experimentally determined model parameters they're using and convert them into unitless parameters. So the user has to describe things like the actual dimensionality of the discretization and then translate their model parameters into those terms.
All right, thanks.
Thank you for your talk and your time, Dr. Sago. We're now going to move on to our next talk.
Right. Thank you very much again.
So our next speaker is Dr. Steve Andrews. Dr. Andrews is a programmer at the University of Washington's Department of Bioinformatics and a research affiliate at the Fred Hutchinson Cancer Research Institute. He has worked on modeling cell biology for 20 years. In pursuit of understanding spatial organization at the cellular level using detailed biochemical simulations, Dr. Andrews developed SMALDEN, the topic of today's talk, which is now the most widely cited particle-based simulator. Please join me in welcoming Dr. Andrews.
Great, thank you. It's a very old picture of me, but that's okay. It's kind of funny to see it. Well, thanks for everybody being here on the very last talk of this session of this school. Show my screen here. Are you seeing my screen? I put it in the.
Yes.
Good. You're seeing the right correct mode.
And there's the actual slides full screen. Yeah.
Great. Thank you. Yes, I got a bit of an introduction here on spatial modeling in general, then on to SMALDEN, my software. And too much clutter on my screen here. Subtitles. What subtitles? All right, I turn off subtitles in this thing. Options are on for subtitles.
What platform is this?
I think
PowerPoint.
they're on.
Oh, I'm
I
off,
can
though.
see.
I think I'm off.
Okay.
I turned off subtitles.
I didn't know how you set them off. I didn't even know you could put them on.
I didn't either until it said I must do it. And I said, sure.
Go to the next slide. Maybe it's not there.
All right. Anyhow, it'll just be a little extra clutter in your screens. All right, I'll try again. Here it is. Oh, off. Good.
Oh, that's gone. Yeah.
Sorry about that. So, please ask questions in this talk. Again, it's been a lot of talks for a lot of days, so I know you're probably tired of talks at this point, but jump in, ask questions. The goal is a somewhat shorter than my full hour talk, and hopefully more interactive than it could be. But I'll jump in with questions if you want to. So the focus has been primarily in this summer school on traditional reaction network modeling of here's a metabolic system or other systems, but the focus primarily is on ODE models for lots of good reasons. ODEs are easy to use, lots of theory. There's flex modes, molecular control analysis. There's lots and lots of good theory, software, tools, everything. There's a great system, and it's often adequate, but not always. So one sort of extension to ODE models are stochastic models. So, as we've seen before, we have A goes to B, deterministically has an ODE and decays exponentially, or stochastically you have many trajectories. So you have individual molecules reacting one at a time, different timing. So does this matter? Depends. If you have a lot of molecules, it doesn't really matter that much. If you have, its variation is 1 root n. So you have 1,000 molecules, probably low importance, percent variation. You get down to hundreds or tens of molecules, it definitely matters a lot. That's a stochastic model or not. So the extension you're going to add to ODE models is stochasticity. Doing so adds a lot of complexity, and primarily you lose most of your tools. You can have a simulator still, but you lose all the analytical additional tools. But that adds realism if needed. This is a pretty picture of very spatial things in biology. A huge fraction of experimental biology is microscope work, is spatial. People looking at all kinds of things inside cells, and it matters. I always like to say a well-mixed cell is a dead cell. So biology is just full of cell organization. And so does this matter, and is it worth studying? So we saw this in the last talk as well. And does it matter? So I have two answers to that. One is yes, if we're addressing spatial questions. Like here's E. coli cells, we call them MIN system. If we look really carefully, see these fluorescent proteins go back and forth in the cells. So pole-to-pole oscillation. And that helps the E. coli find the cell center. So yes, it's a spatial question, and just summation of that. And so to study that question, you build a spatial simulation that then creates back and forth oscillation and helps understand how it works. Or number two, space matters if dynamics arise from spatial inhomogeneity. So maybe things start out homogeneous, but over time things become inhomogeneous. So you get this kind of spontaneous pattern formation. And so then obviously space matters. You want to see those things. So I want to have a high level view of what's out there for spatial simulation. So there's a whole range from 0.1 nanometers with atom size or less, up to 10 microns, 100 microns, cells and tissues. And here's a quick overview of the software that's available. For the most part, I'm missing some things. The small scale, you have Ready and Spring Salad. I'll have more slides on these things. They can do molecules, proteins, dozens of proteins, up to maybe 10 nanometers. Still pretty small. Larger size scale, we got MyWorks Malden from nanometer to 10 microns roughly. MCell is similar. Cetazim is a filament simulator. I'll discuss more later on. Spatiosite is intriguing. That's a lattice-based simulator. And here's VCell as a PDE solver. So that's back to the differential equation approach. And that's now a bit larger, 100 nanometers on up. Also mentioned as well, the larger size scales are Compass Cell 3D and other kind of bigger things. So that's overview. Next slides go a bit more detail here. On the small size scale, 0.1 nanometers, is now pretty tiny for most system biologists. So yet we have molecular dynamics. There's what chemists think of as computational spatial simulation. This is all atoms or maybe single molecules, added solvents. A lot of high computational effort for a very small volume of space, a very small amount of time. That's really chemistry, I would say, or physics. In biology, Ready and Spring Salad are two simulators that do proteins are spheres. They have realistic potentials, or they have little springs between them, and they interact, move around. You can now get protein shapes, macromolecule shapes. You get diffusion of proteins. This shows some crowding with Ready. So pretty small size scales and pretty short simulation times. But those are good tools to use for those things. A bit larger, Spatiosite's an intriguing program. It is a fine lattice with 1 molecule per lattice site. So you can sort of see it here. And it's extremely fast. I was using it a while ago, many years ago, and it was much, much faster than anything else that people were using in my group. But the challenge is, though, this lattice has potential for introducing lots of artifacts. Getting reaction rates to be correct with that is hard. Diffusion is pretty easy, but getting interactions with reactions and surface interactions, those rates are hard to do correctly. I think I'll hopefully solve this point, but that was hard to get. And it still has a smaller size scale because you have the size of 1 molecule times your dimensions to the 3rd power. So you get huge memory demands for large systems. Next scale up is 210 microns, Mark-Smaldon, and M-Cell are very similar. Both are particle-based simulators, which is a continuous space simulation and particles that diffuse in the space. When they collide, they react. And that can hit surfaces and do things surfaces. Smaldon is a bit better physics than M-Cell. M-Cell is much better graphics. So here's M-Cell graphics. Also intriguing at the same size scale is Cytosim. That's a filament simulator by Francois Nedelec in Cambridge. And it does not do molecules or reactions, but he has amazing filament simulations. There's the actin network on a cell. I guess as mitotic spindle portion of that. So first is filaments. Cytosim is a great tool for that. Larger yet, back to lattices, we have MesoArtD and URDME, both at the same university, Uppsala, Sweden. In this case, it's a coarse lattice. So you have many molecules per lattice site instead of one molecule. And then you do a stochastic simulation of as many molecules in each lattice site. Differences are MesoArtD is cubic lattice, URDME is unstructured mesh. Here's a picture of the URDME, URDME. It's an unstructured mesh of the simulation, and each single color is one mesh region. Also larger high scale, virtual cell. It was a PDE simulator initially, so it was deterministic and looked at diffusion and the reactions with differential equations. And this is a V-cell picture. V-cell has grown to become a multiple solver software. It has Smaldon in there, for example, for particle-based simulation, has ODEs, has stochastic ODEs, or stochastic differential equations, lots of things in V-cell. Of course, we mentioned about computational 3D, which is also lattice-based and a little bit larger size scale. So that's better for tissues, which most other things here are better for single cells. So, any questions on this quick survey of software? So here's a summary of a paper I wrote a couple years ago that summarized the main tools for kind of small scaled. So M-Cell Smaldon, it mentioned EGFRD. That's a much more elegant algorithm for doing particle-based simulation. It's much, much faster if you have very, very few molecules, but much, much slower if you have a lot of molecules. So for example, at runtimes, what took Smaldon 22 seconds took GFRD 13 days. So it shows a simulation that was a lot of molecules in it. Also at 9 months for spring salad or 30 minutes ready. So again, these times are relevant for that simulation, but the real essence is these things over here, spring salad ready, are designed for much smaller systems at finer timescales. That's really the differences. All right. So back to the middle size scale, there's got two approaches to getting this middle size scale, which, by the way, I sort of use a term here, but dislike because middle size scale could be anything depending on the field you're in. But in this field, middle scale often means kind of one cell size scale. Approaches are go for this coarse lattice approach and add in detail. You start with the PDE, you add stochastics, then it's called a spatial approach, and you add in more and more detail and get down to this cell size scale. Or vice versa, start with a microscopic picture of everything in there, and then start cutting stuff out. You have MD, you cut out a lot of stuff, cut the solvent, get bright dynamics, you cut out the molecular details, turn into spheres, you get particle-based simulation. So same size scale as cell size scale roughly, but different approaches. And I approached it from this bottom size going upwards. And the question is, does this method matter? So this is a predator-prey simulation which ran with non-spatial on the top, spatial on the bottom, and said, does the simulation method affect the results? The answer is absolutely yes. The classic result is this little oscillation of predator and prey offset by a quarter cycle. If you change simulations, you get quite different results. And in particular, this particle tracking one, you get this kind of boom bust pattern instead of the alternate oscillations. So because it's cool, that's some advice for you guys. One is use the least accurate method that's going to solve a problem, because always when you add in more detail, things run slower, are harder to use, harder to debug, more parameters, more challenging. And so the goal is to solve a problem. So least accurate method you can do that solves a problem. I very often get emails from people who want to use Malden to do ODE problems. My advice to them is, please don't use ODEs for that. And the secondly is establish software. Another pet peeve I have is seeing people rewrite my code or other people's code and say, here's my interpretation of existing code. And what happens always is, it's a lot of extra work. And the end result is not well debugged, not validated, and not as good as the established software. So this was advice here. We keep track for a long time of source citations, Google Scholar. Just try and see what do people actually use out there. Now, this is not actually what people use. This is what people cite, which tends to be kind of name recognition and what people want to put in their papers. But it's really seeing a few tools get most citations, and a whole lot of things are out there as well with first citations. So Capacity is easy to use, graphical interface, good software. There's not special simulator. Though NetGen is the main tool to use for rule-based modeling for complexes. And then down here, Tellurium Roadrunner. So they're low down, but they're great programs as well. So definitely gonna support them and hope they are higher. Advantages of Tellurium Roadrunner are the interfaces, so you can use them at a higher level of detail than Capacity. You can get in there with code interfaces, which you can't do with Capacity. But anyhow, it's just fun to see what's out there for some citations of these simulators. In the spatial world, there's a lot lower numbers, citations per year, but VersaCell, Malden, MCell are widely used. MetroD, URDME, or Leno, whatever. There's a whole lot of things with presentations. So this is not that big a deal, but it's kind of fun to see what people cite. So on to Malden. Again, questions for anybody? Okay. This has been many, many years of work, obviously, so I have papers on it. And the overview is Malden is a continuous space spatial simulator, has fixed time steps, tends to be 10 milliseconds roughly, but can use much smaller, much larger time steps. Molecules are individual spheres, and membranes are thin surfaces. So here's before, this is the E. coli min system. These are single dots or single molecules fusing around. The wire mesh shows the surface of the cell. It's conceptually pretty simple. Molecular actions are diffusion, adsorption, desorption, etc. from surfaces, and molecules react upon collision. Plus a whole lot of features. Well, the overview of features is accurate and pretty fast. Real-time graphics, lots of tools for observing and manipulating the system, robust modeling, free open source, and other sort of features. So, whoops. What it's used for is kind of two tended classes of things. My vision first was system modeling. And here's somewhere there for dendritic spine modeling. Here's E. coli chemotaxis, E. coli min system for cell division and chemotaxis, yeast cell signaling, apoptosis. So various cell systems, it tends to be 5 to 20 chemical species in these things. It tends to be sub-micron to 10 microns. Or then, what I not envisioned initially was a lot of biophysics modeling. So people want to have maybe 1 or 2 species and look really in depth at how crowding affects reaction rates, or crowding affects diffusion coefficients. Or look at diffusion barriers. How does diffusion affected by this little neck on this cell division? Or if you have called hop diffusion, as a particle diffuses and it gets stuck on various interfaces, how does that affect things? DNA target finding, again, as a single species, transcription factor diffusing around finding DNA. So this really happened because I worked really hard making Smaldon quite precise. So it doesn't have good graphics, but for biophysics, it does a good job. So user's point of view, you can download it from the Smaldon website. Installation's pretty easy, hopefully. There's a manual for it, instructions. It's pretty straightforward to download and install. Lots of example files, and then to run it, the interface is a text interface. So you use the terminal interface or the command prompt, whatever it is, and just type in Smaldon in your file name. User's view is a creation file, some text file, put it in Smaldon as a black box, and then as Smaldon runs, it produces graphics and text output, which just as files, and these then get sent into your favorite software for analyzing files. This file, I'll show you with a quick summary. This is the same file from last one that gave that pre-place simulation. You define the graphics, you define things like dimensionality as 3 dimensions here. You call it number of species, rabbit and fox in this case, predator prey. Define your boundaries, your simulation, time start, time stop, time step, colors for species, create some molecule lists, and then diffusion coefficients, put in your reactions, initial condition with number of molecules, and if you want, output files for where to save the stuff. The Smaldon's kind of main core stuff, it's 1D, 2D, to 3D space. Again, continuous space. Time is fixed time steps. There's no built-in units, which is often a challenge to beginners, but it's not that hard to get used to it. So you convert things from your typical value into, say you want to use micron millisecond, and here's microns and milliseconds for different things. Again, there are entities or molecules and surfaces. My goal is to add into that filaments as well in the next year, 2 or 3, but for now it's molecular surfaces. Molecules are points or spheres. Surfaces are these lines here, smooth and thin. And surfaces are built from panels. So you might say, add a circle or add a line or add a triangle, that sort of thing. They have compartments also from surfaces. You can say, the inside region is that compartment. Molecules can be in solution or bound to membrane. It actually has 4 membrane-bound states called front, back, up, and down. But it doesn't matter, actually. The reality is there's other solution molecules or membrane-bound molecules. Procedure is pretty simple. Molecules diffuse, do surface interactions, do reactions, increase the time value, and then observe and manipulate the system, and then back to the beginning. So briefly, diffusion is quite simple to simulate. Things just have Gaussian distributed displacements and diffuse. Afterwards, you address collisions if need be. It happens both in solution and on the surface. They also can drift with a net mobility in one direction, or can do diffusion anisotropically as well. For particle tracking, there's a lot of features for this. So Smooth doesn't work, unfortunately. Oh well. Here are labeled lipids on a cell membrane, and they diffuse around on the cell membrane, and they can hop occasionally from like corral to corral. So this has been done a fair amount in experimental work, as well as simulates very well as well. So you can watch these diffuse and then analyze experimental work with this simulation. And there's, again, substantial support for the tracking simulations. Reactions have been a major part of the core and a big challenge for me. The basic idea is simple. Reactions are two reactants, green and red. When they collide, they react and form blue. The challenge from my work was what constitutes a collision? What's the binding radius that leads to the correct reaction rate? And the answer turns out to be this graph here, which a lot of work many, many years ago, but that now leads to the correct reaction rates. If it's reverse reaction, it gets more complicated. This product then diffuses for a while and then separates and add an unbinding radius here. Things don't come apart in the same location. They have some initial separation. The reason for that is because I want to control the probability of rebinding, because it can go from here back to a immediate rebinding called a recombination, or it can go apart and separate and diffuse away separately. So this is a major focus for a long time. That was many years ago. More recently, I've worked a bit more on diffusion limited reactions, activation limited reactions, more so the chemical physics of getting reactions simulated really accurately. And the small output I should mention, lots of detail about reaction details, but rate constants, the time, binding radius, the chi value, that's activation limited reactions, step length, binding radius, that sort of stuff. So lots of detail here if you care about it. Again, it's important to me for a long time, so I'm very careful to validate reaction rates against theory. So straight lines theory, the dots are experiments. And here's the spread. The spread also matches what it should match. First thing in order. And then going on to volume interactions. So in this simulation, you have particles in a box, and they have a sort of volume. To do that, I have two algorithms. These molecules collide after diffusing, and then there's an overlap. One algorithm is trying to push them apart by the overlap. So they got to come apart by the same much that they initially overlapped. Or else you back up the time a little bit to where they first collided, then reflect off that position. So you back up a little bit here, reflect off that reflection plane. Both of these are very accurate, and they lead to quite accurate volume, which I checked using hard sphere fluid. This is a well-known chemical physics system of put a whole bunch of spheres in a little box and then measure the sphere-to-sphere separation on average. So this shows there are no spheres close by because there's extruded volume, and then there's a initial shell of spheres surrounding each one, and then all oscillations. Again, the line is theory, and dots are simulation. So good agreement. I worked some on looking at crowding. So again, have the very small size scale detailed simulations about physics. So my question was, how does crowding inside a cell affect the reaction rates? So here's a picture of reactions occurring in a crowded system. So we know crowding affects diffusion coefficients, slows diffusion down, obviously, but it also reduces the volume available for reactions, so they go faster. And so the combination of these things change the reaction rates. Again, small has been a good tool for that. On the surfaces, a lot of interactions are possible. Here's a surface, here's molecules. They can reflect off surfaces, transmit through surfaces, absorb into surfaces, adsorb, desorb, transmit partially with some reflection, some transmission, or a surface-bound molecule can hit another surface and interact there. So lots of inputs the user can give that says how to make surface interactions as you want them. or can have on-surface diffusion. Molecule is on the surface and diffuses, and then this looks at how do you do it accurately. Well, it has to go from here to here, and then go to here, and bounce off the edge. So it has to work out, took a bit of work, but works out very well. It can do quite large time steps and do it accurately with large time steps. So the blue line, the blue dots show one long time step, goes here to here in one step. The gold dots show many small time steps, and the solid line shows theory. It's all the same, obviously. Compartments, basically you can define compartments in essentially the obvious way. A compartment is a region inside a surface or outside a surface, but a region bounded by surfaces. And you always have a interior defining point. Say, this point is inside the compartment. So any point in here that's inside the surface and closest point is interior by definition. If you're over here, you're cross surface from that point, therefore you're external. So, as expect. Newish development is you can now have surface compartments can diffuse. So here's diffusing molecules inside a compartment, diffuse molecules on the compartment surface, and the compartment as a whole diffusing as well. Also, this can now defined over here. I think it does at some point. Yep, these now bind together. And the final part of the basic loop in the program is observations and manipulations. So there's actually it's a great, I call it a hack part of the code. You can add in anything you want to that's not part of the core. So you can observe, say, I want to observe molecules, or observe molecules in a compartment, or track molecules, or whatever. Just plan to observe all kinds of things with a simulation. Or you can manipulate things. So this is the hack part. You can say, it's completely physically unrealistic, but I want to fix molecules somewhere at some concentration. Or I want to add in molecules somewhere. There's a syringe here saying, now dump in a bunch of green molecules right here. So a huge wide range of manipulations are possible. Graphics are none is possible if you want something to run fast. OpenGL good if you want better quality graphics, or better, let's use that for publications basically. But fast graphics obviously run faster and are often adequate. Also VTK is possible as well for well, these are real-time graphics run during a simulation. VTK is done afterwards. The major topic is protein complexes. So lost proteins don't function as individual proteins, but they complex together in these large multimeric protein complexes. For example, in the yeast's primary response system, STE5 is a scaffold protein. STE5 binds to STE4, which binds to STE18, that's G protein. It also binds to STE20, STE11, STE7, FUS3, and in all possible combinations of these things. And there's phosphorylations on these proteins here. So how do you deal with that huge list of possible proteins, possible species, really, plus all the reactions of all those species? So one solution is automatic generation of species and reactions from much rules that say how they are formed. So two methods here. One is BioNetGen has solved this problem many years ago. So Smallton now uses BioNetGen directly. You can give a Smallton BioNetGen file and say, build the species from this BioNetGen file. So here's another example. This is a protein complex. A receptor can bind to a ligand and or to messenger protein. But messenger protein, it can get phosphorylated. Phosphorylated messenger protein. Here's a reaction network. So you can create this if you want to with BioNetGen. Or a new thing is as a wildcard system in Smallton, which is fun to write. So here it says ligand in solution plus receptor bound to anything, R star, like wildcards in your operating system, binds to LR star. So LR plus anything. Or star R plus M star forms star RM star. So it's like wildcards in an operating system, and that will kind of R star means R with anything beyond it, but also in addition it means the anything is copied over into the product. It's kind of a neat addition to wildcard systems of identifying stuff here and copying it over to the product, expanding the system.
Is that literal text-based asterisk things here?
Yep, it's all text-based.
Okay. So if you name your species appropriately, then this sort of thing will work.
Exactly. So the challenge actually, and it's kind of fun actually, it's a fun challenge, is to come up with a good naming scheme. So this scheme is pretty easy, saying, well, my system, my letters are L, R, and M, and P for phosphorylation, and I create this network with these letters, and it all comes together with a text-based wildcard system. Yeah, thanks. So with this new scheme, I built a whole bunch of other networks. Here's just other simple networks, polymerization with breaking in half, transmission translation. There's again MIND system. What this is, but more systems. There's transposase, that's what it is. But it's really fun to build these different systems with wildcards, and it's surprisingly powerful. Another feature is hybrid simulation. Often you want particle-based simulations and part of the space where you want detail. Then you want lattice-based simulation where you want less detail. So left has particle-based, right side is lattice-based, and now it's called a port in between where molecules can cross freely. And so that gives now much more efficient simulations with a kind of the lattice-based part. Whoops. Another addition is libsmalden. So at first this was a C and C++ interface of Smalden. So you can get set parameters and get parameters translations through the C interface. And now the new addition is a Python interface. Here's the full code in Python, which builds a simulation. It says, create a simulation, add in red and blue species, and then the reaction is, let's say, B plus R, our substrates, reacts to form R plus R, rate 20. But the key is the blue molecules diffuse, red molecules don't diffuse. And so it forms this growing cluster in the middle. And so this is all Python code, which runs the Smalden through Python. So Python is built on top of the C interface, it's built on top of Smalden. And then a bit more advice here. There's a whole parameter output in Smalden because, again, the focus is on precise biophysics. I give the user full information about what's going on. Say, here's all the reactions, all the details about reactions, all the molecules, everything in there. And my advice to users is, at first, don't bother. But as you get into Smalden more and more, make sense of these outputs and learn to use them. My last slide, since you have very few questions. Here's some information about Smalden. Website is smalden.org. It has a GitHub site, of course. Read the docs. And then various papers, kind of two more important ones. And then please ask questions now or later on. I'm always available to answer questions about Smalden. And then thanks to lots of people. So that's my end of my talk, and I've lots of time left over for questions if you want.
Yay, thanks.
Thank you.
I suppose one question I have is, so you've got, is there any connection with SBML?
Nope. I'd like to, but it's done currently.
Okay, so that's something that could be done possibly.
Yeah.
Okay.
Because a very small part of Smalden is the species and reactions. That's a core part of it. but it's actually
Yeah.
a small part of the total. Kind
Sure,
of
it's a small part. Yeah.
Yeah. So I'd love to have spatial SBML. I mean, that's been discussed for a long time. I'd love to
Yeah.
have SBML with the spatial stuff additional.
Have you looked into that at all? The one that currently exists? I don't know
Mm,
whether it's over-engineered or not.
yeah, a
It's-
while ago
yeah,
it was over-engineered and not really appropriate for Smelton.
It's all, I mean, everything that's there, we could, it might be useful to just to chat about it. It's been a while since I've worked directly with the spatial groups and stuff, but it's probably, my guess is that the stuff you want was there.
Yeah. The challenge
Although it
is
doesn't, like, the only thing is that it doesn't do moving. Like the spatial part can't move, like it's not dynamic.
right. That's one challenge. But it there's quite different representations of facial stuff. Lattice-based versus not lattice-based.
That's the problem.
Yeah.
Yes.
And just find it's been so hard to get the same language for the different representations.
Isn't it better to perhaps just describe the biology and then let the particular engine
Now you need to describe the space somehow.
generate the appropriate mathematical?
Yeah, but there's no biology that says like it's a 3x3 box. That's not
right.
But couldn't you just say this is a 3x3 box? It has these dimensions. You implement it in any way you want.
Yep, and you can do that with spatial.
Yeah, I worry that'd be
Uh,
work actually pretty easily with Smelton, but it'd be hard to implement lattice-based simulators from that description. So they would not really.
It's got to be a description that's completely independent of the final implementation.
Yeah.
I mean, so the different ways of describing space are, the idea is that you pick the one that makes sense for you and you do that. And then if you are feeling ambitious, then you can write converters from one type to another. And the converters won't maybe be perfect, but it'll at least let you write your thing.
That's the right way to do it.
Yeah. That was the original, like, Jim Schaff's group, like, plan. I mean, partly, again, like we were talking about how it's hard to get everyone to agree on a subset.
Yeah.
That was his solution to it's hard to get everyone to agree to a subset, was we'll define the different ways and let people write what they want and read what they want. And then it turned out that like some people wanted to read and write combinations of some of those. Like they want like a grid for this part and a mesh for another part, or I don't, or like defining like you can define the sphere, what do they call it, CSG, like solid geometry, CSG. I don't remember what the C stands for, but solid geometry, basically like spheres and cubes, and like morphed, that you can morph in different dimensions and stuff.
Yeah.
So you can define it that way, or you can find a mesh of triangles, or you can define a, like, based on a picture, because different situations require different things. But then if the hope is that if you have a picture, but you have a simulator that runs on triangles, you just sort of triangulate your picture and move it on or something.
I have a question. How does this differ from the like molecular dynamic simulations or the particles in there?
It's much coarser. So most MD stuff is single atoms with interatom potentials, or maybe single molecules with intermolecular potentials. And it's short, small size scale, small time scales, and you have potentials between atoms or molecules. You have solvent often in there. So a lot of detail. I'm ignoring all solvent. I ignore molecules I don't care about. So same like the min system. I have min D, min E, and that's all. I'm ignoring all the other molecules in the system, other proteins as well. So it's much, much coarser in terms of the treatment.
Yeah.
Okay, thank you.
You wouldn't be able to do it otherwise, would you? I mean, you can barely model a protein in enough time, let alone if you try to take that to the whole cell. Yeah.
Yeah.
Okay. Okay. Any more questions? Otherwise, we're coming to the end of the entire week, I think. Is that right? There's nothing else, Veronica or Janice? Nothing else?
No.
No?
This is it.
That's it. Well, thank you very much, everybody, for attending.
Putting the link to the survey form in the chat again, just if you haven't filled it out, and a bunch of you have, just give us feedback about what you thought.
We can keep the Slack channel